Welcome, and thank you for taking the time to watch the following presentation on Transfection 101. My name is Seth Hansen, and I'm a Research Technology Specialist for MilliporeSigma. During this webinar, I will briefly review the basics of transfection. I will also discuss the various methods of transfection and the parameters to consider during your transfection experiment. Afterwards, I will review the various transfection reagents that are available on sigmaaldrich.com. To kick things off, let's begin with the basics of transfection. Transfection is the process that introduces DNA or RNA into eukaryotic cells. It is a common and essential method to study and modulate gene regulation, which allows for a better understanding of gene function and protein expression within the context of the cell. Transfection can be used for analysis of regulation of gene expression and production of recombinant proteins, making it a critical link between the study of genomics and proteomics. There are a few primary types and methods of transfection, which I will go over in the next few slides. The two types of transfection used experimentally are known as transient and stable transfection. In transient transfection, the nucleic acids introduced into the transfected cell are not permanently incorporated into the cellular genome. Therefore, the effects of the nucleic acids within the cell last only a short amount of time. Often, transient transfection is used for pathway analysis and promoter or functional studies. In contrast, stably transfected cells permanently integrate nucleic acids into their genomes, and thus the effects of the nucleic acids can be investigated over a long period of time. Stable transfection is typically reserved for research that absolutely demands it, such as protein production on a medium or large scale, or gene therapy. There are multiple ways to get DNA into a cell. Physical methods include electroporation or direct microinjection. Viral-mediated transfection is primarily based on adenovirus, lentivirus, and retrovirus. Particle-based transfection via nanoparticles such as gold is another method used to deliver DNA into cells. The most common transfection methods are chemical-based, which include the use of calcium phosphate, DEAE dextran, and PEI, as well as liposomal and polymer-mediated approaches. Electroporation is a technique that involves the exposure of cell membranes to high-intensity electric pulses. This causes a temporary destabilization in certain areas of the cell. During this transient destabilization event, the cell membrane becomes highly permeable and allows the entry of DNA. Electroporation is an easy, non-chemical technique that can yield high transformation efficiencies in various cell types. Although this technique does not alter target cell morphology and functions, the method can cause cell death if transfection is not done under optimal conditions. Viral-mediated transfection involves the use of viral vectors to deliver nucleic acid into the cells. Viral delivery systems such as lentiviral, adenoviral, and oncoretroviral vectors can be used for transferring nucleic acids even in hard-to-transfect cells. Although viral delivery methods are highly efficient, they can be quite laborious. Moreover, most viruses require containment and careful monitoring of biosafety levels. Before performing viral transfections, it is also important to consider several limiting factors such as the lytic nature of viral vectors, cell line packaging, and host cell specificity. Particle-based transfection via nanoparticles is another method of transfection. Cationic gold nanoparticles can be processed to form a complex with plasmid DNA, which is then transfected into cultivated cells. The calcium phosphate transfection technique involves the precipitation of DNA and calcium phosphate. The precipitation is facilitated by mixing a Hepes-buffered saline solution with calcium chloride solution in DNA. Glycerol shock is often used to enhance the DNA uptake in certain cells. While this technique is cost-efficient and can be used for transient or stable transfections in a wide range of cells, relatively small changes in pH can affect the efficiency of transformation. In addition, it is essential to maintain reagent consistency for reproducing the assay results. This transfection method does not work in RPMI or other media with high phosphate concentrations. Liposome-mediated transfection, or lipofection, techniques involve the use of liposome-forming cationic lipids, or non-lipid polymers. Such lipids and polymers form liposomes that associate with DNA and merge with the cell membrane to enable the intracellular transfer of DNA. This technique can be used for efficient delivery of nucleic acids into the cell cultures without much difficulty. Lipid transfections can also be adapted for cost-effective as well as high-throughput systems. However, the transfections are usually cell type specific. This table outlines the various transfection methods discussed in the previous slides. Each method has key advantages and disadvantages that are important to consider when planning a transfection experiment. The most important point, however, is that no single transfection method is ideal for all projects. Now that we have covered the basic methods and types of transfections, I will go over the parameters to consider during transfection. There are two main components in the process of transfection: the nucleic acid transfection complex and the cells in which the complex is set to enter. When preparing the DNA or transfection complex, it is important to consider the purity, amount, dilution medium, and ratio of the nucleic acid. The types of reagent, timing, and amount of complex used during transfection is also critical to the success of the experiment. In addition, the type, density, quality, and passage number of the cell being used is also important to consider. These factors influence the outcome of your transfection, including the amount and quality of protein achieved. Transfection efficiency is another vital parameter to consider. Transfection efficiency is greatly influenced by the cytotoxicity of the transfection reagent used. For example, given a scenario where 100 cells are in a well, if 90 cells die and 8 of the surviving cells are transfected, the transfection efficiency is 8%. When using a transfection reagent with low cytotoxicity, such as extreme gene reagents, of the 100 cells, only 10 die, and the remaining cells continue to grow to 150 cells total. 75 of these cells are transfected, leading to a transfection efficiency of 50%. This scenario demonstrates the key role cytotoxic effects have during the transfection experiments. Reagents with minimal cytotoxicity can greatly improve transfection efficiency for your experiment. Now that I have provided a complete overview of transfection, I will move on to our transfection portfolio. We are the exclusive distributor of Roche reagents, including the Extreme Gene family of transfection reagents. There are 4 key products in the Extreme Gene portfolio. Extreme Gene 9 and HP are for transfection of DNA. The key difference between these reagents is that Extreme Gene 9 delivers DNA to commonly used cell lines, while HP is excellent for difficult to transfect cells. The Extreme Gene 360, launched in early 2019, offers efficient delivery of DNA, siRNA, miRNA, and Cas9 RNP complexes into all cell lines, ranging from common to difficult. In addition, Extreme Gene HP and 360 are both free of animal-derived components. All Extreme Gene products show high cell survival, minimal cytotoxicity, and have simple protocols with easy optimization. This image provides a visual of the simplicity of Extreme Gene reagents. Combine the Extreme Gene reagent with your nucleic acid into a single tube to create your transfection complex. Then incubate at room temperature, and the mixture is ready to be added to your cells without having to change media. In addition to Extreme Gene reagents, we also offer Gene Juice, another differentiated family of transfection reagents. Gene Juice is a DNA transfection reagent uniquely formulated with novel protein and polyamine. It has minimal cytotoxicity, is compatible with or without serum media, and is ideal for high-throughput transfection in multi-well formats. It works in a wide variety of cell types with very high transfection efficiency. The data shown here demonstrates Gene Juice's superior transfection efficiency compared to commonly used competitor reagents in 6 different cell types. It is very easy to use with no media changes needed between steps and flexibility in experimental design. In the side-by-side comparison experiment shown here, COS7 cultures were either left untreated, transfected with gene juice, or transfected with a popular cationic lipid-based transfection reagent. In these images, taken 48 hours post-transfection, cellular damage can be observed as the rounding up and detachment of cells from the plate surface. This demonstrates that gene juice is less cytotoxic than other commonly used lipid-based transfection reagents. Finally, we also provide other specialized transfection reagents specifically designed to transfect DNA, protein, peptides, or siRNA for different cell types. This table outlines the various product families within our transfection portfolio. For each product, the corresponding cell types, transfection complexes, applications, and highlights related to these products are shown to assist you in the transfection reagent selection process. And with that, I would like to thank you for taking the time to listen to the Transfection webinar. I hope you now have a greater understanding of the transfection methods and products offered. If you have any questions, please feel free to either contact us via the information listed on this slide or visit our website, sigmaaldrich.com/transfection. Thank you and have a great day.